My lab broadly focuses on understanding genotype to phenotype relationship. We are using cutting-edge long-read sequencing, combining it with functional genomic dataset to understand how genetic variation or mutations in the genome that are found naturally in natural populations cause functional changes. When we find a mutation, we don't really know often what the mutation does to the organism. And we use CRISPR-Cas9 to engineer that mutation into a particular genetic background. And then we try to find out what that mutation is doing at cellular level, at tissue level, and at the overall organismal level. We are not really tied to any organism. We are currently using Drosophila melanogaster for our model organism, but we have collaborators who are working with cancer cell lines in mouse models, mosquitoes. We also have goals in expanding our work in mosquitoes. We are focusing on Drosophila right now, but we're not tied to it. We try to venture out in other organisms if the research demands it. We follow the standard practices. Depends on who is leading the work. The person leading the work generally gets the credit by being the first author. If it ever occurs that two people equally contributed or will be equally contributing to a manuscript, we'll usually talk about that before the manuscript is written so that everyone knows that they are going to be co-first author, or one person is going to be first author, other person is going to be second author, and so on. And these are generally mutually agreed upon before the project develops fully, so that there's transparency. I try to maintain an open conversation about authorship as the manuscript or the project develops. There is a decision made in the early stage of the project, but it's kept open and more organic as the project develops. I meet with students formally every week. I try to maintain an informal communication open, so my door is always open. It's just an extra layer of flexibility I live with students. So if they want to talk to me, they can talk to me anytime they want. One thing I tell the first day is, what is it that you want? If you think that academia is the right call, you can continue, thinking that way and go on the next phase of training. But at any point, during grad school or after grad school, during postdoc, if you feel like, well, this is not what I want to do, I don't really want to become an academic scientist, I want to do something else, then that is the right call. And I try to support them whatever way I can. I mean, if they want to go for an internship, sure. If they want to go to a specific conference, that would help them build their skills or network to move them into that direction they want. Yeah, absolutely, I'll support them. One thing that I try to do in my lab, I try to my lab a little on the smaller side so that I can invest in the career success of my trainees.